Well, this blood's just been removed from a radial artery. It's therefore been removed from the systemic arterial circulation. And as we can see, it's 98% saturated with oxygen. And this is what we would expect from arterial blood, 96% or more saturated with oxygen. This means that of all the hemoglobin molecules in this blood that you are looking at, 98% of them are carrying the full amount of oxygen that they can carry. Or to put it another way, it means the lungs have been working with 98% efficiency, which is really quite impressive when you think about it. Now, the reason the blood is bright red is because it contains oxyhemoglobin. It contains a lot of oxyhemoglobin and only a very small amount of deoxyhemoglobin. And when the oxygen combines with the hemoglobin, this is what gives rise to the bright red colour. Oxygenated blood, carrying oxygen from the lungs to the tissues where it is required. And this is also important clinically. So if you see a wound and you see this bright red blood in the wound, that indicates there is arterial hemorrhage going on, because the bright red blood comes from the arteries, and that might guide your management, because it will be a good idea to restrict the amount of arterial hemorrhage. Well, this blood has just been removed from a systemic vein, and as we can see, it's quite a lot darker than the previous bright red systemic arterial sample that we've just looked at. The reason it's darker in colour is it contains a higher proportion of deoxygenated hemoglobin. That is a higher proportion of deoxyhemoglobin, sometimes referred to as reduced hemoglobin. So when you see this sort of dark coloured blood in a wound, it indicates that there's venous hemorrhage going on. And it's actually quite interesting when we take blood from a venous sample and we put it in our blood gas machine, we find that the oxygen saturation is still around about 75%. So what this means is that when the arterial blood, the systemic arterial blood, goes to the tissues, it's not giving up all its oxygen by any means. It's only giving up about 25% of its oxygen. And that partly deoxygenated blood is returning to the heart, to return to the lungs, to be reoxygenated again. But when it returns to the heart in the systemic veins, and then when it's pumped from the right ventricle of the heart through the pulmonary arterial system, it's still about 75% saturated, not fully deoxygenated. Blood consists of two basic components. The first is the fluid component, referred to as plasma, or sometimes referred to as serum. And within the plasma floats the second component, the blood cells. And blood volume is normally made up of about 55% plasma and 45% cells in men. In women, it tends to be about 60% plasma and 40% cells. And if an anticoagulant such as heparin is added to a sample of uncoagulated blood, which is then centrifuged or allowed to settle for quite a while, all of the cells will settle out on the bottom of the tube while the plasma remains floating on the top. And that is what we see in this picture. We can see the clear straw-coloured plasma at the top and the red cells at the bottom. And actually this sample is not that well settled, not that well centrifuged, because you can see that that volume of plasma is not 55% like it says on the annotation, but in actual reality it is, because this is not a fully settled sample. And if you look just above the red cells and just below the plasma, you can probably see a thin layer of white cells. These are the leukocytes, or the white cells. So this is what you see when you anticoagulate and centrifuge a blood sample. The plasma on the top, the thin layer of white cells and then the thicker layer of red cells. And the proportion, or actually the percentage of blood which is made up of red cells, is described as the hematocrit. So the hematocrit typically in men would be 44–45%, and in women typically the hematocrit would be around about 40%. Plasma is a yellowish fluid which is about 91% water, and this keeps the blood fluid so it can circulate freely around the vasculature of the body, allowing normal circulation. The rest of the plasma is made up of various substances in solution and suspension. So it includes plasma proteins, nutrients, dissolved gases, waste products. And a lot of the biochemical tests that we do on blood are looking at the biochemical composition of the plasma. Plasma carries absorbed nutrients that have been absorbed from the gut to all tissues of the body which need them. It's the universal transport system. These include nutrients such as glucose, amino acids, vitamins, and minerals. The fatty component that's actually absorbed from the gut goes into the lacteals first in the villi, and from the lacteals it goes through the lymphatic system and actually drains into the subclavian veins, particularly into the left subclavian vein, as chylomicrons. So it's actually absorbed in a slightly different way. But once it's in the blood, it's still transported around in the plasma. When tissues metabolize protein, especially if the protein is deaminated, broken down to release energy, then that generates waste nitrogen. And this nitrogen goes into the tissue fluids and it goes into the plasma and it combines with water, and that forms ammonia. And ammonia is a very smelly, very alkaline, very toxic molecule. So the body doesn't want ammonia floating around for long. So what happens is, as soon as the ammonia circulates through the liver takes out the waste ammonia and converts it into urea. And urea is much less toxic, but still highly soluble. And once formed, the urea is transported in solution from the liver to the kidneys, and the kidneys incorporate the urea into urine and thereby excrete it from the body. Proteins are organic molecules. They contain carbon, hydrogen, oxygen, nitrogen, and sometimes other elements as well. And there are three main plasma proteins circulating with the blood plasma: albumin, globulin, and fibrinogen are the main ones. And proteins are very large molecules, and so their presence is vital to generate plasma osmotic potential. Without this osmotic property, the blood would be unable to reabsorb tissue fluid into the venous end of capillaries. Albumin is the most common plasma protein and also generates most of the osmotic potential of the plasma. The globulin proteins include the immunoglobulins, and you might remember that immunoglobulins are antibodies which allow the body to fight off infection. These are the immune proteins, and without these we'd probably die of the next viral or bacterial infection that we pick up. Other globular proteins act as molecules which transport some hormones, and minerals around the body. Fibrinogen is a plasma protein which is essential for the process of normal blood clotting. Now, as fats are not soluble in water, they're actually transported around the body bound to plasma proteins. And these combinations of fat and protein are termed lipoproteins, and they're soluble in water. Endocrine glands produce hormones, the chemical messengers, and these are released directly into the bloodstream. So endocrine glands have a good perfusion of blood. There's lots of blood going through the endocrine glands. And when the hormones are released, they are released directly into the blood. And there they circulate to get to the target tissue where they're going to have some physiological effect. So obviously the endocrine hormones are going to be transported in the blood because they're released directly into the blood when they're made, transported in the blood to the target tissue where they're going to work. The two main forms of blood cell are the red cells, or the erythrocytes, which is the biconcave disc you can see on the left of this picture. The second main group of blood cells are the leukocytes, or the white cells, and there's an example of one of those on the right. And the third cellular component in blood are the platelets, or the thrombocytes. And these aren't full cells, they are cell fragments. They derive from large cells called megakaryocytes. So they are cell fragments, so they are part of the cellular component, and they are essential for the process of blood clotting. The reason blood is red is because it contains the red blood cells, the erythrocytes. And the function of these red blood cells is to carry oxygen from the lungs to the tissue, or all the tissues of the body. And they also carry some carbon dioxide from the tissues back to the lungs to be excreted in the exhaled air. Now, the percentage of blood which is composed of the red blood cells is termed the hematocrit. And the hematocrit can be lowered in some forms of anemia. And we can see from this picture that the red cells are small biconcaved discs. And the reason they're red is that they contain the haemoglobin oxygen carrying pigment. And each molecule of haemoglobin in the red cells can carry 4 molecules of oxygen. So we see that the red blood cells are biconcaved discs. And this shape provides a large surface area for gaseous exchange. So there's more surface area for the oxygen to get into the red cell. Compared to its volume, it has a large surface area. And as well as that, some of the capillaries in the body, for example in the kidneys or in the brain, are actually smaller than the 7 micrometer diameter of the red cells. And they have to deform and squeeze to get through the smaller capillaries. And this biconcaved shape gives them flexibility so they can deform to squeeze through small capillaries. So as we've said, the red cells are about 7 micrometers in diameter. That's 7/1000th of a millimeter. And in normal blood, every cubic millimeter of blood, every mm⁻³, every cubic millimeter, contains about 5 million of these red blood cells. So here we see views of a red cell from above on the left and from the side. From above, the outer thicker layer of the cell is going to appear darker red than the thinner inner layer. Almost like the middle of a donut, but it's not a hole, there is actual continuation of the cell. And the lighter area in the centre of the erythrocyte is called the area of central pallor. Pallor means paleness, representing the thinner middle. And this should be a third or less of the total diameter of the red cell. Erythro is the prefix that means red. So erythro is red, and of course cytes are the cells. And the erythrocytes in circulation in the bloodstream don't have a nucleus. Now, when they were being formed in the bone marrow, they were nucleated, but they lose that nucleus at around about the time they are released from the bone marrow. So in circulation, they have no nucleus. So the red cells are the only cell in the body which has no nucleus. They had one in the past, but they haven't got one when they're circulating. We've said there's about 5 million per cubic millimetre of blood, and they're about 7 micrometers in diameter. And the reason they're red is because they contain haemoglobin. Haemoglobin is the actual pigment that carries the oxygen. And a single haemoglobin molecule can form a loose bond with 4 oxygen molecules. And although the haemoglobin molecules are large, at the centre of them, they are based on 4 atoms of iron. And so if there's a deficiency in the diet of iron, if people do not get enough iron to eat, then the haemoglobin can't be properly synthesized and they can get anemia. So the mature red blood cells circulating in the blood are unique because they don't have a nucleus. And this is good because it allows more space for carrying more haemoglobin and therefore increases the oxygen carrying capacity of the cell. But it also limits the lifespan of the cell because the cell is not able to repair itself because it's not carrying the genetic material from the DNA in the nucleus. And for this reason, they have a limited lifespan of round about 17 weeks or 120 days. But these are absolutely vital, the red cells. In the lungs, the dark red deoxygenated haemoglobin will combine with oxygen to form the bright red oxyhemoglobin. And in the systemic capillaries, the reverse will happen. Oxyhemoglobin will give up the oxygen molecules and revert to being deoxygenated again. So in a sense, the haemoglobin molecules are like little trucks. They carry oxygen from the lungs and they drop this oxygen off in the tissues. And it's the colour change which accounts for the bright red blood seen in systemic arteries as opposed to the dark red blood seen in systemic veins. The erythrocytes are formed from blood stem cells found in the bone marrow, and this process is called erythropoiesis. And the red bone marrow is found in flat bones, for example in the bones of the skull, the bones of the sternum, the ribs, the pelvic bones. Red bone marrow is in the flat bones, and it's also at the ends of the long bones, in the epiphyses, the end part of the long bones. So thinking about bones like the radius and ulna, humerus, femur, tibia, fibula, all have red bone marrow in the ends of the bones. And these contain the stem cells to make the new blood cells. And a stem cell is any cell which is capable of differentiating into other cell types. Now, at the end of the lifespan, the old red cells are phagocytosed by large cells called macrophages. They're eaten up and digested by macrophages. And this is good because it means that the proteins which were in the old red cells can be recycled again. So it's quite an efficient process. And this process takes place mostly in the spleen because there's lots of macrophages in the spleen, but it also takes place in the liver and the bone marrow to some extent. So the proteins from the old red cells are broken down into amino acids, and the iron from the haemoglobin from the old red cells is transported back to the bone marrow to be recycled to make new red blood cells. So there's no waste in this process. But there is a coloured pigment from the haemoglobin that's not broken down, and this is converted into the bile pigment called bilirubin, which is taken up by the liver and excreted into the small intestine as a component of the bile. And of course, bile is useful because it emulsifies fats and it colours and deodorises to some extent feces. Well, the reason that the red cells are red is because they contain this red iron-containing pigment called haemoglobin. And this is the oxygen-carrying molecule in the erythrocytes. And because it's a combination of protein and iron, protein and a metal, it's called a metalloprotein. So it's the oxygen-transporting metalloprotein. And actually, it also carries about 10% of the carbon dioxide, which is produced in the tissues back to the lungs as well in the form of carbaminohemoglobin. So carbaminohemoglobin is when the hemoglobin is combined with carbon dioxide. But of course, we're normally thinking about hemoglobin as carrying the oxygen. The deoxygenated hemoglobin becomes oxygenated and forms the bright red oxyhemoglobin. And typically in the systemic arteries, we're going to get saturations of 96 to 99%, although it can easily go up to 100% if you take a few deep breaths. Now, someone's once calculated that there's 640 million molecules of hemoglobin in a single red blood cell. Well, I'm not quite sure how he counted them. But that's what they say, 640 million molecules of hemoglobin per red cell. And hemoglobin is an impressive molecule. As well as carrying the oxygen, it can collect the oxygen and it can give it up very quickly. So when the oxygen is travelling through or when the red cells rather are travelling through the pulmonary capillaries, the oxygen comes into them really quickly. In about 1/100 of a second, the hemoglobin molecules can oxygenate as the blood passes through those pulmonary capillaries. So men, typical hemoglobin values, as we see here, 13 to 18 grams of hemoglobin per 100 mils of blood. Women are a little bit less. And 1 molecule of hemoglobin is going to carry 4 molecules of oxygen. And this means that in normal levels of hemoglobin, 100 mils of blood can transport 20 mils of oxygen. So it's the red coloured pigment in the red cells, carries the oxygen. It's what makes the blood red. Hemoglobin which is not carrying oxygen is sometimes called deoxyhemoglobin or reduced hemoglobin. It's the same thing. And this hemoglobin has the potential to loosely bind or bond with molecules of oxygen. And as we've said, each hemoglobin molecule can carry 4 molecules of oxygen. And this forms bright red oxyhemoglobin in the lungs. And this bright red oxygenated blood is going to drain via the pulmonary veins into the left atrium of the heart. So the pulmonary veins are carrying this oxygenated blood into the left atrium, down into the left ventricle, When the left ventricle contracts, the blood is going to be pumped out into the aorta and into all of the systemic arteries of the body. The systemic arteries are going to feed the blood to the arterioles, and that's going to feed blood into the capillaries. And it is the capillaries which are the actual site of gaseous exchange. In all the other blood vessels, the walls are too thick for there to be any significant gaseous exchange taking place. So in the tissues, the oxyhemoglobin will dissociate, releasing the oxygen which will diffuse through the wall of the capillary into the tissue spaces, into the cells to supply the cells, specifically to supply the mitochondria, to facilitate the process of respiration in those cells. That will leave the deoxygenated hemoglobin or partly deoxygenated hemoglobin at the venous end of the capillary. That will then drain into venules, into systemic veins, into the inferior and superior vena cava, which drain back into the right atrium. From the right atrium, the blood will go through the tricuspid valve into the right ventricle. It will be pumped out of the right ventricle into the pulmonary arteries and go to the pulmonary capillaries. And again, it is the pulmonary capillaries where the processes of gaseous exchange take place. They're the only vessels where the walls are thin enough. So carbon dioxide will pass from the blood that's derived from the pulmonary artery into the pulmonary capillaries. The carbon dioxide will pass from the blood into the alveoli, and oxygen will pass from the alveoli into the blood. Then again, the blood will drain from the lungs in oxygenated form, going back to the left atrium, completing the circulation of blood with the oxygenation and deoxygenation of the hemoglobin molecules. Now, in the arterial blood, you might find that the oxygen saturations are 97, 98%, something like that. If you take a few deep breaths, it can easily go up to 100%. And in the veins, the oxygen saturation goes down typically to about 75%. So it's not that the hemoglobin is completely reducing as it goes to the tissues, it's only losing about 25% of its oxygen. And it's going to the lungs to be topped up again. So typical oxygen saturations in systemic arteries are going to be in the high 90s. Typical oxygen saturations in the systemic veins, therefore the oxygen saturations in the right side of the heart and in the pulmonary arterial system are going to be in the order of 75% saturations. Leucocytes are the white blood cells. Leuc means white, cytes, cells. And their basic function is to protect the body against infections and infectious disease. And this will include resistance to bacteria, viruses, fungi, protozoa, parasitic worms, and indeed some toxins as well. Some leukocytes will also attack and kill some cancer cells. And they also, unfortunately, defend the body against transplanted organs. And this is because white cells are able to distinguish between self and non-self material. And they will attack anything they perceive to be non-self, not part of the body. The white cells are much less numerous than the red cells. Each cubic millimetre of blood contains about 7,000 leukocytes. Now, there are two main forms of white cells which are described according to the appearance of their cytoplasm. Some white cells have cytoplasm which appears granular, so this group are classified as granulocytes. Other white cells have cytoplasm which appears clear, with no obvious granules, and these are called agranulocytes. A or an, of course, means without. So these cells are without granules. Of course, this is a very simple way to look at things, and it's based on classification using light microscopes. It's what you would see with an ordinary light microscope. There are three main types of granulocytes, and these all end in "phil": neutrophils, eosinophils, and basophils. So the phils are the classic granulocytes. And there are two main forms of agranulocytes: lymphocytes and monocytes. However, the thrombocytes, that is the platelets, are also normally included in this group of agranulocytes with the lymphocytes and the monocytes.
Now this graphic is designed to give us an overview of the structure of the blood. So at the top we see there's the blood divided into cells and plasma. And we can see the water contained in the plasma and protein contained in the plasma, nutrients, hormones, waste products are in solution in the plasma. So that's the fluid component. Then as we go to the left, we see that the blood also contains cells. And we see that this is red cells, which are the erythrocytes, and also white cells. So the white cells can be divided into granulocytes, and these are the ones that end in "phil". So the basophils, neutrophils, and eosinophils are the granulocytes. And the white cells, the leucocytes, that is, leukocytes and white cells are exactly the same thing. They can also be divided into agranulocytes, cells which with the light microscope did not have obvious granules in their cytoplasm, although they do with the aid of an electron microscope. But what we call the agranulocytes are divided into monocytes, lymphocytes, and the thrombocytes are the platelets. And when the monocytes migrate into the tissues, we start to call them macrophages. But the lymphocytes themselves are divided into large and small lymphocytes. The large lymphocytes are the natural killer cells, the NK cells, and the small lymphocytes are divided into B and T lymphocytes. And the T lymphocytes are themselves subdivided into helper, cytotoxic, and suppressor T cells. So it's the role of the B lymphocytes, the small B lymphocytes, to synthesize the immunoglobulins, to make the antibodies that combat infection in the process of acquired specific immunity. And the T cells stimulate this process, and the suppressor cells say when this process has been carrying on for long enough, they limit the length and magnitude of the immunological response. And the cytotoxic cells are capable of directly killing cells. And they can kill cells which are infected with viral particles, and very often they can kill malignant cells as well. So it's a schematic to give us an overall feel for the components of the blood. Well, it's important to remember that the white blood cells were named in the days of light microscopes only, where it wasn't possible to get that much microscopic detail on what we were looking at in the cells. So at that time, some cells were described as being agranulocytes, and this means they did not have obvious granules in their cytoplasm. So they were agranulocytes, A meaning without. So the cells that were without granules, or at least apparent granules on light microscopy. But there are other types of cells that were called granulocytes. And there's three main types of granulocytes: neutrophils, eosinophils, and basophils. And we notice that all of these granulocytic leukocytes, all of these types of white blood cells, end in "phil." We'll be looking later on at the two main forms of agranulocytes, which are the lymphocytes and the monocytes. However, on this slide, we are looking at the granulocytes. And I think we can notice that the neutrophils have got the smallest granules. Then the eosinophils tend to have bigger granules, and the basophils have the largest granules of all. Now, there's very few basophils actually that you will see on blood slides. Most of them seem to migrate into the tissues where they form the mast cells. So the granulocytes, remember, neutrophils, eosinophils, basophils, they all end in "phil." Well, if you start looking at blood slides, it would take you quite a long time to come across one field where there's 4 neutrophils in view, because the neutrophils, of course, are very much less common than the erythrocytes, than the red cells. But I think you can clearly see the red cells in this picture, and you can see the area of central pallor in many of the red cells that is much thinner than the area around about the outside of the cell. And also we can see some small blue stained cellular fragments dotted around this picture. These are the thrombocytes, or the platelets, that we can see. But I think you can see from this why neutrophils are sometimes called polymorphonuclear cells. Poly means many, morph, shape. There are many shapes in the nucleus, typically 3 or 4. So for example, this cell on the bottom left, we can see the lower left neutrophil, we can clearly see that there are 3 lobes in the nucleus, joined by nuclear strands. And actually, as the cells get older, they can grow more lobes in their nucleus. So an old neutrophil may well have 5 lobes in its nucleus. So we can see that they are polymorphonuclear cells. Now, the neutrophils are the most common form of leukocyte. That is the most common form of white blood cell, normally comprising about 60 to 70% of the leukocytes in the blood. And most of the granules in the cytoplasm are fairly small and actually might be difficult to see under a light microscope. And the granules in the cytoplasm contain enzymes such as lysozyme and other enzymes which are capable of digesting bacteria in the process of phagocytosis. So these neutrophils are capable of phagocytosing bacteria and dead body cells. And the nucleus of a neutrophil has 2 to 5 lobes, as we've said. So it can be as little as 2 when the cell is young, connected by fine threads. So they're sometimes called polymorphonucleocytes. Now, the key thing is during an infection, the number of neutrophils will increase significantly, because it is the neutrophils that help the body fight off the causative organisms of the infection. And this is particularly the case with bacterial infections. And this increase in number of the neutrophils is called a neutrophil leucocytosis. An increase in the number of white cells is a leucocytosis. And as well as killing bacteria, neutrophils can kill fungal infections as well. And in addition, they'll phagocytose dead or damaged body cells. So when body cells are killed in a disease process or as a result of injury, it's important that they are phagocytosed and removed. This means the amino acids and the cellular and the chemical components they are composed of can be recycled to make new tissues, which is good. But if they're left in the tissue, they'll form what's called necrotic areas of dead tissue. And these will supply food and habitat for bacteria, increasing the likelihood of infection. And neutrophils migrate out of inflamed blood vessels into areas where there's injury or infection to perform these immunological functions. So if there is an inflamed area, the neutrophils will be able to migrate out of the capillaries into the tissue, where they can phagocytose dead tissue cells, where they can phagocytose infecting bacteria, thereby providing a vital immunological function. So if you find that the number of neutrophils has increased greatly in the blood, this may well be because there is a bacterial infection. Now, the eosinophils are often slightly larger than the neutrophils, maybe 13 to 15 micrometers in diameter. And the cytoplasmic granules in eosinophils are larger than those seen in the neutrophils. And normally only about between 2 and 4% of the leukocytes in the blood are eosinophils. The nucleus in an eosinophil normally has 2 lobes, although looking at this one it appears to have 3. But the lobes are joined by a thin nuclear thread. And eosinophils help the body fight infections of protozoa. So you might think of amoebic infections, for example, like amoebic dysentery or amoebic liver abscesses. The eosinophils help fight the protozoa, these large eukaryotic cells that are parasitic. And eosinophils also fight larger parasitic worms and other parasites as well. And if a parasite enters the body, eosinophils attach themselves to the surface of the parasite and stick to it using adhesion molecules. And they then deposit digestive enzymes and other toxic molecules from the granules inside the eosinophil onto the parasite. And these granules contain digestive enzymes and other nasty things that the parasites don't like. And this toxic assault from the eosinophils will weaken or kill the parasite, reducing the damage it's able to inflict on the body and hopefully reduce its ability to reproduce. And eosinophils are also attracted to areas where allergic reactions are occurring, such as in the bronchioles in people suffering from asthma. And once in the area where the allergic reaction is going on, they're able to chemically neutralize and detoxify some of the inflammation-causing substances, such as histamine, which are involved in the generation of the allergic inflammatory response. And this is why eosinophil numbers increase in parasitic infections and in allergic disorders. And many eosinophils migrate out of the blood to protect tissues against potential infections. And this often happens where body surfaces are exposed to the external environment. So they can get bacteria and things from the external environment. And this is why eosinophils can be found in association with mucous membranes in the digestive and respiratory tract, as well as in the female reproductive tract. And if there's an increase in the number of eosinophils, we will call that an eosinophilia. And if there's an eosinophilia, this can indicate parasitic infection, amoebic infection, or allergic reactions such as asthma. The basophils are the least common of the leukocytes. Only 0.5% or less of the leukocytes will be basophils. And the nucleus is irregular and lobed. But it's often hard to see the nucleus because there's so many large, abundant granules in the cytoplasm, they obscure your view of the nucleus. Now, the granules in the basophils contain histamine, heparin, and bradykinin. And the physiological role of basophils in the blood is not particularly obvious, but it is well known, of course, that heparin prevents blood coagulation because it's an anticoagulant. But most basophils actually enter the tissues where they remain localized. And in the tissues, they're called mast cells. And their main function is to facilitate inflammatory reactions in response to insults of the tissues by releasing their histamine and these other inflammatory mediators into the tissues. So if a tissue is damaged, the mast cells will release inflammatory mediators. And this will generate the inflammatory response that we normally expect to see: heat, pain, redness, swelling, and loss of function. So here we see a basophil that's migrated into the tissues. And when they migrate into the tissues, they change their name. They're no longer basophils, they become mast cells. So really what we're looking at here is an ex-basophil, now called a mast cell. And these tend to stay in the same tissues for long periods of time. And they facilitate inflammatory reactions, and especially they can generate allergic inflammatory type reactions. So on the cell on the right here, we can see the nucleus, and we can see the very large granules containing the histamine and the heparin and the bradykinin. And when the cell's stimulated to release these inflammatory mediators, it's almost like the whole cell falls apart. It granulates, or degranulates rather. So in the picture on the top left, we can see the granules degranulating, and they all fall out of the cell, locally releasing these inflammatory mediators, bringing about a localized inflammatory reaction. And from this graphic in the top left, it almost looks like the cell would die as a result of this. It seems to be falling apart, but it doesn't. It releases the granules in this degranulation process, and then the cell will survive and recover again after this. So the mast cells were basophils, but they now live in the tissues as mast cells. Leucocytes are the white blood cells, and a leucocytosis is a significant increase in the number of white blood cells in the blood. And as we know, it's the leukocytes that are involved in immunological function. So if there's an increase in the number of neutrophils, this indicates bacterial infection, because the neutrophils combat bacterial infection. For example, they can phagocytose them. And an increase in the number of neutrophils would be a neutrophilia, indicating a bacterial infection, because the number of neutrophils would increase as the immune system seeks to combat the bacterial infection. An eosinophilia would be an increase in the number of eosinophils, indicating parasitic infection, amoebic infection, or disorders such as asthma. Basophils form mast cells, and if there was an increase in number of basophils, that would be called a basophilia. But that's not a clinically common situation. Now, if there was an increase in the number of monocytes, that would be a monocytosis. And as we'll see shortly, the monocytes are involved in combating chronic bacterial infections, such as tuberculosis. So in tuberculosis, you would expect a degree of monocytosis. And the lymphocytes are there to combat primarily viral infections by making antibodies. So a lymphocytosis would probably indicate viral infection particularly. And this is why we do differential white cell counts. Yes, we're looking for a leukocytosis, but is it a neutrophilic leukocytosis? Or is it a lymphocytic leukocytosis? Because these would indicate different sorts of likely etiological infections. remember that there's basically two forms of lymphocytes. There's large lymphocytes and there are small lymphocytes. The large lymphocytes are the natural killer cells that will kill cancer cells, that will kill cells infected with viruses, with viral particles, the natural killer cells. But the small lymphocytes can be B or T type lymphocytes. And this is a small lymphocyte that we're looking at here. But by looking at it, I can't tell whether it's a B or a T type lymphocyte. But it is a lymphocyte. So the large lymphocytes are the natural killer cells. In this picture, we see one of the small lymphocytes that can be B or T lymphocytes. But I don't think you can tell the difference by looking at them. It's more a chemical sort of difference or histological type of difference between the B and the T cells. But the lymphocytes are the most common form of a granulocytes, normally making up about 20 to 25% of the circulating leukocytes. And most of the lymphocytes are classified as small. And you can see they are quite small. They're not a lot bigger really than the red blood cells which are surrounding it. And you can also see some platelet fragments off to the right there, the thrombocytes, the smaller cell fragments. And the nucleus in the lymphocyte is large and fairly spherical. And large numbers of lymphocytes can be found in lymphatic tissue, such as the spleen, the lymph nodes, the tonsils, and the appendix. And there's two main forms of small lymphocytes. There's the B lymphocytes and there is the T lymphocytes. And the difference is that the B lymphocytes mature in the bone marrow, while the T lymphocytes mature in the thymus gland. And the function of the B lymphocyte cell lines is the production of antibodies. These are the immunological proteins, the immune proteins produced in response to the presence of specific antigens in the process of acquired immunity. And an antigen, of course, is something the immune system recognizes as being foreign, such as a measles virus. So the lymphocytes are making antibodies in response to exposure to antigens. They are making antibodies which are immune proteins, the immunoglobulins, that will combat antigenic infections, such as viruses or such as bacteria. On this slide, we notice a neutrophil on the left, and with a large purple-stained nucleus, we see a large lymphocyte, one of the natural killer cells. And having said that the lymphocytes are agranulocytic cells, we can see granules in the cytoplasm of this lymphocyte. Now, this doesn't actually change the classification. Still, technically, lymphocytes are classified as agranulocytes, but they still have granules in their cytoplasm that contain digestive enzymes and things that can destroy virally infected cells or malignant cells. So the large lymphocytes are somewhat granular, and they are the natural killer, or the NK cells. So to summarise the lymphocytes can be large lymphocytes, or there can be small lymphocytes. The large lymphocytes are the natural killer cells, and the small lymphocytes are the B and the T lymphocytes. It is the B lymphocytes that synthesize the immunoglobulins, that is, make the antibodies, and T helper cells, T suppressor cells, and T cytotoxic cells
are the three subdivisions of the T lymphocytes. And we've said there's a lymphocytosis that can indicate viral infections. So there's an increase in the lymphocytes that can be viral infections. For example, you might get a lymphocytosis in viral hepatitis, but you could also get it in measles, mumps, toxoplasmosis, and some chronic infections as well. So it's not necessarily viral infections. There's other possible causes of a lymphocytosis.
Now, when there's an infection, this means that foreign material will enter the body. And on the surface of this foreign material, there are particular molecules that the immune system recognizes as being foreign. And these are called epitopes. And there's many millions of different types of foreign material. Epitopes can be introduced into the body in many types of infection. And you have individual B lymphocytes that recognize different antigenic epitope type molecules. And when a small group of the B lymphocytes that recognize a particular antigenic molecule bind to that molecule, when that molecule is present, when the antigen is present, that antigen will bind to molecules that recognize that antigen, recognize that epitope on the surface of the B cell. And what the B cell will then do, once it's recognized the antigen, is that B cell will divide many, many times. This is a process called clonal expansion. So a clone is something with the same genetic material. So this stimulated B cell that's stimulated by the presence of the antigen will divide many, many times, producing a huge population of B cells, all identical. That means you get a huge population of B cells that can recognize this foreign antigenic epitope and will produce the immunoglobulins that are specific to countering that particular infection. This is a specific acquired immunity. And the huge population of B cells that derive from the original stimulated B cell, the factories, if you like, for the antibodies, are the plasma cells. So millions of plasma cells will be produced that are all identical. They'll produce the same antibody, that is the immunoglobulin. That antibody or immunoglobulin will then counter the infection and eliminate the infection. And this is cellular immunity, because it's the plasma cells, which are the descendants of their ancestors, the original B cells, that are producing all these immune proteins to get rid of this infection that's been introduced into the body. So when there's an infection, when we're making lots of antibodies to counter an infection, you'll get populations of these plasma cells, giving rise to a lymphocytosis, an increase in the number of lymphocytes. And then when the infection has gone, there's no need for all these plasma cells producing lots and lots of antibodies, so the population will greatly reduce again. But there will be a few thousand B memory cells left, and these will stay in the circulation probably for the rest of the life of the individual in many cases. And that means if the individual is exposed to that infection again, they'll have several thousand memory cells that can start dividing into another population of plasma cells really quickly if the antigen is experienced again. So that means that a secondary immune response can be mounted much more quickly than the primary immune response. And indeed, with these secondary immune responses, people can be exposed to the antigen for a second time, and the antibody response due to the plasma cell clonal expansion is so rapid they don't even realize that they've been exposed to the antigen and don't get sick at all. So plasma cells, absolutely essential for producing large volumes of immunoglobulin to counter specific infections. So here we see that there are two main classifications of small lymphocytes, the B and the T. and there are three forms of T lymphocytes, and these are T helper lymphocytes, T cytotoxic lymphocytes, and T suppressor lymphocytes. So these different sorts of T lymphocytes. Now the T helper cells help the B cells or stimulate the B cells to produce antibodies. So it's only the B lymphocytes that will produce the antibodies, but they will only do so when stimulated to do so by a T helper cell. And this is why HIV, human immunodeficiency virus, can cause acquired immunodeficiency syndrome, AIDS, because the HI virus, the human immunodeficiency virus, invades and kills T helper cells. And this means that they are unable to stimulate the B cells to produce the antibodies. And without the antibodies, the person becomes immunodeficient. Now, as well as stimulating immunological responses, it's also important that the degree of the immunological response can be regulated. So when B cells have produced enough antibodies to deal with a particular infection, the T suppressor cells inhibit the B cell lines, and antibody production will therefore be reduced. And this is important if you think about things like autoimmune diseases, where the immune system will attack the body's own tissues, which is not at all what we want. Now, the T cytotoxic cells have got a range of defense functions also. They're able to detect and kill cells which are infected with viral particles. Now, although it might sound a bit drastic for the body to kill its own cells, if a cell is infected with viral particles, it will kill the viruses inside the cell and so prevent them from infecting further cells. And cytotoxic T cells are also able to detect and kill some cancer cells. So it may be that malignant cells arrive fairly frequently in the human body, who knows? Maybe every day we develop cancer. But God willing, we won't get that condition because the cytotoxic T cells will recognize it at the 1 or 2 cell stage and then eradicate the malignant cells before a tumour can be formed. And T cytotoxic cells can also attack foreign material directly, such as parasites, in a similar way that we've already learnt that the eosinophils can do. And if there's an increase in the number of the lymphocytes, this is a lymphocytosis. And this will mean that more antibodies have been produced to counter an infection, normally caused by a viral infection, but it can occur in other infections, such as tuberculosis, often with an increase in the number of monocytes as well. So a lymphocytosis, we're thinking about infection, probably a viral infection, but also possibly some sorts of bacterial infection.
This picture shows the amazing complex nature of the external cell membrane surface of a T lymphocyte. With a light microscope, you really don't get any idea of this level of complexity. And this healthy T lymphocyte will help the B lymphocytes to produce antibodies.
So here we see one of the large monocytes with their typical kidney-shaped large nucleus. And these are classified as a granulocytes, but with good microscopes these days, you can actually see small granules in the cytoplasm. So they are large phagocytic cells. And if they start eating things, they can grow to being very large indeed, even larger than this one that's shown here. So typical kidney-shaped nucleus. And the key thing about the monocytes is that especially if the capillaries are inflamed, they can migrate through capillary walls. And then in the tissues, they can move around by this process of amoeboid movement. So they can patrol through the interstitial spaces. An amoeboid movement involves the flow of the cytoplasm within the cell, rolling the cell forward. And once in the tissues, monocytes change their name and they become macrophages. And if any bacteria or viruses are encountered in the tissue, they will be rapidly destroyed by this process of cell eating, the phagocytosis. So in the tissues, they become macrocytes. Macro means big, and phage means to eat. So they are macrocytic, big cells, macrophages, big eaters. So in the tissues, we will call them macrophages, literally big eaters. And the number of monocytes is particularly likely to increase with chronic bacterial infections, giving rise to a monocytosis. Well, here we see a macrophage that has been actively eating by this process of phagocytosis. And as we can see, it's become positively huge. You can see just next to it the ordinary sized red blood cell with its area of central pallor to give us a sense of scale of how big this macrophage has become. And the macrophages are derived from monocytes. And the thing about the macrophages is they're found in body tissues. And indeed, there are some macrophages in most body tissues. Sometimes they're fixed to tissue cells, and sometimes they patrol around a tissue. And once in a tissue, the macrophages can live for many years, just waiting in case there's an infection or they need to perform some immunological function. So the macrophages are one of the first lines of defense. They're in the tissues, just waiting in case there's a problem that needs their attention. And they'll phagocytose infecting viruses, bacteria, and they'll also phagocytose necrotic tissue, which might have been killed as a result of trauma or infection or a disease process. That's good because you don't want necrotic tissue left lying around in the tissues, because bacteria can eat the necrotic tissue as a metabolic substrate. And also the necrotic tissue is a good habitat for bacteria to live in, so it's best to get rid of it. And as well as that, healthy cells can't migrate effectively over necrotic tissue, so for many reasons it needs to be got rid of, and the macrophages will do that by phagocytosis. Now, as well as directly phagocytosing any invaders, any viruses or bacteria that the macrophages themselves come across, they're even much more clever than that, because they can release chemicals called cytokines. So if an infection is encountered, they'll release a chemical messenger called a cytokine. And the cytokine will circulate to the bone marrow, where it will increase the release of neutrophils and other immunological cells to help fight the infection. And monocytes also produce pyrogens, which act on the hypothalamus to increase body temperature during infections. And these functions mean the tissue macrophages act as an early warning system, and they can initiate localized inflammatory responses and the systemic inflammatory response syndrome as well. And the monocyte macrophage cell system is in the body used to be called the reticuloendothelial system. So the monocyte macrophage cell system is the term used to describe all of the monocytes, fixed and mobile macrophages in the body. And we used to call that the reticuloendothelial system. We now call it the monocyte macrophage cell system. Well, here we see a macrophage actively phagocytosing. Now, these long strings that look like strings of pearls are actually bacteria. Now, a cockeye means that the bacteria is round. So we can see there's lots of individual round bacteria here. So we know we're dealing with a cocci. And the bacteria that occur in strips like this are Streptococcus. So what we see here is lines of Streptococcus. This is how Streptococcus appears. It occurs in these long strips. And the macrophage is actively phagocytosing them, taking them into its cytoplasm. Once the bacteria are in the cytoplasm of the macrophage, they will be surrounded by membranes from lysosomes, and digestive enzymes will chemically digest and dissolve away these bacteria, thereby killing them. And we also see a fairly good sense of scale on this picture. So 5 micrometers is that line along the bottom. So we can see that the macrophage is about 10, 15, maybe 12 micrometers across and maybe 20 micrometers long. And we can also see the individual scale of the bacteria. These look like they're about half a micrometer in diameter, maybe a little bit more.
So once the bacteria have been consumed into the cytoplasm of the macrophage by phagocytosis, lysosomes containing lysozyme and other digestive enzymes will fuse with that vacuole and will digest the bacteria. Now, monocytes, as we've said, can differentiate into macrophages, but they can also differentiate into cells which develop rather strange shapes. So here we see this one has spread itself out. Maybe it's trying to detect some antigens. It's covering a large area where it could detect possible infecting bacteria or viruses. Now, as monocytes change in the tissues, they could become macrophages, as we know, but they can also differentiate into dendritic cells. And dendrite means branched. So here we see these dendritic cells have many branches. They're putting tentacles out all over the place. And this means that they can cover quite a large area and can detect any antigens in that area. And for this reason, they act as antigen presenting cells. And then if they come across an antigen, they will take it in and process it, and they will present that to the T cells, which will then initiate the cellular immune response. So they're detecting antigens in the tissues, that is, infections in the tissues, going to the lymphatics and informing the lymphocytes that it's time to mount a specific acquired immunological response, APCs, antigen presenting cell. Having first detected the antigens, of course, they then present them. Now, if we stain specifically for these differentiated monocytes, which are now dendritic antigen presenting cells, we can see that they are guarding numerous body tissues. So here, for example, we're looking at the epidermis. You can see the packed, tight cellular structure of the epidermis. And the dark staining cells there are the dendritic cells that are positioned in the epidermis to detect any antigens, that is, any infection getting into the epidermis, the top layer of the skin. And below this, we can see the dermis, which is the lower layer of the skin. We see there are less cells in the dermis because it contains more extracellular material, such as collagen and elastic fibers. But we can also see that dotted around, just in case, we have these dendritic cells ready to present antigens, to detect antigens, and then present them to the immune system to generate an immunological response. So amazingly detailed, intricate defenses against the possibility of infections. Well, you've got to look at quite a few microscope slides to get a nice picture like this.
But we can see the red cells, we can see the small cellular fragments, which are the thrombocytes or the platelets. On the left, we can see the polymorphonucleocyte, the neutrophil. Then we can see the lymphocyte with its large nucleus in the center. And then we can see the monocyte, large cell with a large single nucleus. So the three main types of erythrocyte there, all present on one rather nice photograph. Now in the circulatory system, of course, it's absolutely vital that blood is a liquid. But if there's a cut, we want that blood to clot to block off the hole in the vascular system, otherwise the liquid blood would simply keep running out. And here we see these yellow strands that are fibrin. Now the fibrin is precipitated out of a soluble protein in the plasma called fibrinogen, and when it precipitates out it forms these long sticky strands like a network, and the red cells will stick to that, and in this shot we even see a white blood cell sticking to that, all being clotted together by the fibrin mesh, forming a blood clot to prevent excessive hemorrhage. Here's another nice picture of a blood clot. We can see the red cells, the fibrin strands, and a couple of white cells thrown in there for good measure. Well, here's my diagrammatic, rather simplified representation of a blood clot. But we can see there's a neutrophil, we can see there's some red cells, and we can see that they're held together by fibrin strands. Now, coagulation of blood or clotting of blood is a complex process, and there are 12 main factors involved in the plasma, and that's why we can see the X11, which is the Roman numeral, of course, for 12. We normally use Roman numerals to indicate the clotting factors. So, for example, you might have heard that haemophilia is deficiency of factor 8. I'm not going to go into it here, but you can see that we need platelet-derived factors, activated clotting factors, and calcium, and that will convert a protein which is made in the liver called prothrombin into thrombin. And when there's activated thrombin around, that will convert fibrinogen, which is a soluble protein, into these strands of fibrin, which is precipitated out, insoluble and sticky, to which the other cells stick to. And vitamin K is required for the synthesis of prothrombin and fibrinogen. Well, here we see a neutrophilia, an increase in the number of neutrophils in the blood as the body responds to an acute infection. In this slide, we see an increase in the number of lymphocytes, a lymphocytosis. And if there's too many lymphocytes, this might indicate viral infection like hepatitis, as we've said. Could be infective mononucleosis, measles, could even be tuberculosis, but very commonly it's a viral infection causing the lymphocytosis. Well, we see two monocytes here together, and we don't see enough of the blood field to decide if this is indeed a monocytosis or just a rather fortunate shot. But monocytosis could occur in an infective mononucleosis, Hodgkin's disease, chronic bacterial infections such as tuberculosis, and other long-term bacterial infections typically. Now in this shot we see an increase in the number of eosinophils and eosinophilia, an increased number of eosinophils in the blood. And this could be caused by allergic reactions such as asthma could cause this, parasitic infections as the body responds to parasites
and possibly in some cancers as well, and eosinophilia. Too many eosinophilic leukocytes. Now here we're looking at a relatively large blood field, and I can only see one solitary leukocyte in the whole field. There's only one neutrophil here. And this is a neutropenia, a lack of neutrophils. And sometimes this is referred to as an agranulocytosis as well, a lack of granulocytes in the blood. And this could occur in some viral infections or tuberculosis, or it could occur in systemic lupus erythematosus or rheumatoid arthritis, possibly in aplastic anemia. Sometimes it occurs secondary to administration of cytotoxic drugs, or indeed anything which will sufficiently depress the bone marrow, and it could be adverse reactions to drugs. An account of white cell count of less than 1 predisposes to infections, and a white cell count of less than 0.5 predisposes to potential life-threatening infections. So definite lack of neutrophils here in this neutropenic slide. Well, here's a few normal hematological values. You do see slightly different quotes by different laboratories depending on who's conducting the assays, but this is a rough guideline as to some normal hematological values. Well, I think we've concluded, or we've given evidence in this slideshow to show that blood is essential for the life of every creature. Thank you for watching and listening.